welcome to the Clinical Trials Methodology course discussion of rigor and transparency. And I am happy to have Will and Dietrich join me. And there's Dietrich, great. So I'm happy to have Will and Dietrich join me. We're going to be discussing a variety of issues, and I'm going to start by sharing my screen here. Maybe, yes. I wanted to introduce, so Will and I are Will is the primary director of the Clinical Trials Methodology course, and I co-direct it with him. And Dietrich is formally from the NINDS, intramural, in charge of the clinical center, and he is currently the medical director for early clinical development at Neurocrine Biosciences, and as Chris Coffey pointed out, probably one of the last people to run a marathon before the pandemic, because he ran the LA Marathon. So you go, Dietrich. So just as we're kind of warming up, first I'm going to ask you to answer a question. Please write your name and your institution, your area of research into the chat box. We are going to be using the chat box for questions. So if you could please access that, I guess.
It's at the bottom of your screen. Just click on the chat icon. Yep.
Yeah. And I'm hoping that people are typing this in.
Yep. Well, we only have 3 so far.
Oh, okay. I'm not seeing any. For some reason, I'm not seeing any.
If you click on the chat icon, Laurie, you'll be able to see what's there.
It may not be visible to her because she's sharing her entire screen.
Oh, okay.
I think there may be some way around it, but I can do a little running commentary.
Okay, great.
Thank you, Dr. Lee from Neuromuscular. Thank you, Dr. Burgos. Thank you, Dr. Shirani in MS. Thank you, Dr. Conwit from NINDS. And thank you from Dr. Tegler from neurology. And I apologize, I'm just gonna default to calling everybody doctor. I know that we may have people from other research domains and other research professionals here.
I'm gonna ask for you to look at the next question, which is, please list an aspect of scientific rigor in your area of research that you would like to have discussed today. And so we're going to start loading some questions, hopefully for that. While you're typing in, I'm also going to say that we have on the panel Robin Conwit from NINDS, who is another co-director, and Chris Coffey from where are you from, Chris? Sorry, University of Iowa, who's head of the data center here, and wishes he'd run the last marathon. So, all right, are we getting comments in there, Will, for scientific rigor questions?
Well, so scientific rigor it can be a tough question. So I think maybe people are thinking about what they want to write.
Okay.
This is a very important part of your grant applications and very heavily weighted by reviewers. So thinking about what you, in your area, like in stroke, you have the STAIR criteria, but sort of thinking, what sort of questions you have, like what thoughts you have about how best, there's just so much research out there that sort of comes before what you're planning to do. Where do you start when you're trying to summarize it? So,
Right. So I'm going to just give a quick overview. So hopefully some of you were able to watch the video for this talk that Will and I gave a few years ago from previous webinar. But in your grant application, you're going to get asked about the rigor of the prior research that supports your grant, and as well as the rigor of your own design. But you want to look at prior research, you want to look at the rigorous experimental design for robust and unbiased results. You want to look at the relevant biological variables, and then authentication of key biologicals. And this is looked at on your grant application. And here is a checklist that kind of reviewers may use to look at this. So, for example, for the general considerations, you can see here, are the results reproducible? Is there a lot of variability when they were repeated in the same lab? Are the findings robust? Can you replicate them, and have they been replicated in other labs? And what you really want to do is you want to address this in your application and talk about what are the gaps or what are the things that support your application. And then the experimental design, you want to look at the preclinical data again, and this is more specifics. And you can see that they're really key questions. Some of them may or may not be answerable, and those are the kind of things that we're going to discuss with Dietrich and Will. And then the translational considerations as you look at taking this to the next step in your clinical trial. So if, does it cross the blood-brain barrier? Is there adequate justification for the proposed dose? All these things are looked at by reviewers as they go through to look at the rigor of your study. And then I'll give you two websites that are helpful. This comes specifically from this website, Transparency in Reporting Guidance, which I want to thank Robin Conwit for sharing this with us. And then there's also a trans-agency activities. This has really been a focus of NINDS since a paper that was led by the director of the NINDS at the time in 2012, really emphasizing the need for preclinical rigor prior to moving into a clinical study. So I don't want to read this whole slide. So I think I'm going to stop sharing my screen, although these will be available. This is all being recorded, so you will get this. Just move to discussing this, openness to discussion, unless Will has something specific. Were you going to show the video, Will, or no?
Yeah, I know that, again, we're not going to necessarily ask people who reviewed the YouTube video, expectedly call them out, but we do have about, it's about 11 minutes. It's being played at normal speed from our webinar that we did back in 2017, so that I can share my screen and we can just show that. And I think that it will go over a couple of the things that Dr. Gutmann just covered, but I think it's good to just review those and sort of pound those in. And one thing that I did put over in the chat box is I try to give like a little bit of an example. It's very important when you're evaluating therapeutics for brain ischemia to ensure that they've fared well under the STAIR criteria and other preclinical experiments. But a persistent issue in terms of me as a consumer of that is that oftentimes there isn't a range of severities that is tested. Whereas clinical stroke, people present with a range of severities. We use treatments in people with a range of severities, but oftentimes some of the animal models that are done fix severity, like I will occlude their carotid for this many seconds to maintain consistency of injury. But we need to learn about how well treatments might work under a variety of of levels of injury, and cardiac arrest is the same way. although it's a little harder for us to tell how big the injury is when the patient presents. So that would be an example. So I think as we play this video, try to think of things from, like known issues, things that you're afraid to ask. You could say, hey, I study a disease that doesn't have any animal models. What do I do? Try to put some things there from your own research, just things that you struggle with when you design your early phase clinical trials, or things that you think are really effective, that you have a lot of good translational and preclinical studies that help guide you. So while we play the video, just kind of put stuff over there, and I will share my screen, and I will share my computer sound, and I will optimize screen share for video clip. All right. I'm gonna make this bigger. And in the YouTube video, my head was in it, but I actually managed to crop my head out of this, so. All right.
By way of background, back in 2003, NINDS had 3 contracts, and the idea was to replicate promising studies in spinal cord injury. And interestingly enough, there were a lot of very promising high-profile studies in the preclinical space, or the basic science space and translational space there, with 18 of those published. But with the methods that were reported in these papers, they were only able to replicate about 10% of the published findings. And I think that something is, one of the fundamental scientific goals of our course is for you to do transparent and reproducible research. At the clinical trials meeting, one of the things that they were talking about is that replication is a little bit more difficult in clinical trials, because a major positive finding of a clinical trial makes it challenging to replicate that clinical trial, given the need to treat future patients with the newly effective treatment. However, the idea of reproducibility and transparency are sufficient details from that clinical trial published such that you could reproduce their methods and feel confident that, given the data and the responses of the patients within that trial, you would be coming to the same conclusions, and that data was collected rigorously. So in the basic science or translational science spaces, replication is quite important. Reproducibility is also important there, but those studies actually can feasibly be replicated, whereas in many of the clinical trial spaces, we're looking for reproducibility primarily. And there was a workshop that was convened by NINDS. The director at the time was Dr. Story Landis, and there's a large group of esteemed authors on this paper, and they really wanted there to be minimal requirements for the translational, the late animal studies regarding how to, we think of the consort requirements for human clinical trials, to really add a lot of those things that improve the validity of these experiments and the reproducibility, including such things as sample size estimation in animal studies, randomization, blinding, data handling, data inclusion and exclusion, stopping rules, and thorough and transparent reporting and accounting for animals and what was sort of going on with each of the outcomes. And this is a great checklist, and often if I'm I refer to it when I'm thinking about working with colleagues who do translational research in animals, so that we're trying to design those studies to meet most, if not all, of these suggestions, because they will help us use that preclinical research. So if we go back to the basics in terms of from the perspective of any scientific experiment, a good experimental design and reporting of it underlies having rigor and reproducibility of these findings. And the idea of what makes a good experimental design is be unbiased, in that there wasn't a as you see the cartoon here, there's a flaw in your experimental design. All the mice are Scorpios. Again, not exactly sure differentially what that would do with the treatment they're testing there, but there may be an impact. Those mice are all too similar. Have a high degree of precision, which means it's going to be, whatever the noise of the assay or the outcome in the preclinical or animal study is, it's going to reliably be measured and not subject to a lot of random noise. Have a wide range of applicability, and basically be simple and have the ability to calculate what the actual effect size is. And these are one of the this is from a classic paper by Cox in How to Plan Experiments. And as any biostatisticians on the call know, many statistics and biostatistics curricula feature a class on design of experiments, and that's a major area. And again, in biomedical science, we weren't all required to take a design of experiments class at some point in our training. So it's important to know this and really be thinking about this when we're designing studies, both in the in the animal and translational arena and in the human arena. So the National Institutes of Neurologic Disorders and Stroke was actually really ahead of the curve in this. I think NIH-wide, many of the reforms and leadership that NNDS undertook have been manifested across NIH through the statement of scientific premise and other aspects that are now routine parts of grant review. And if you could go to this link, it was provided even in the instructions for people who are applying to our course, and this covers several of the major points that we're going through here. But thinking about what when you're designing or proposing clinical studies, thinking about what is the underlying evidence and how well it supports your current application to move forward. So if we go back to the that major paper by Dr. Landis, we can come up with a sort of general checklist, and I'll go into this in a little bit of detail. And again, using the term preclinical here, are these results for preclinical studies, are the models relevant to the clinic, right? One thing that you can commonly see in some stroke preclinical studies is that animals are pre-treated with a neuroprotectant before the stroke occurs. And one of the things about stroke that I think we've all sort of discovered is stroke does occur unpredictably. So unless you're going to put the entire population at risk for stroke on that agent so that it can be circulating in the bloodstream before the incident stroke, that sort of mechanism isn't, that sort of mechanism in the animal is potentially not going to be relevant to the clinic. So this is why there's that need for communication with scientists who focus on animals and scientists who focus on humans that say, well, the earliest I think I could get it into somebody after stroke may only be 30 minutes to an hour or up to 3 hours, 4 hours. So thinking about those aspects, are the results reproducible? Is there variability when it's repeated in the same lab? Again, these are gold standards of what we should be striving for and looking at that research, but it may not always be there. It certainly isn't. And then the robustness of the findings. Have other laboratories using other animal models of disease found similar magnitudes of effect of the proposed treatment? And again, this is what will be helpful to picking interventions that will have the best chance scientifically of going forward. And this checklist here, blinding of treatment allocations, blinding of the raters of the outcome determination, this is good science, either for interventional clinical trials in humans, even retrospective research or cohort studies, or in terms of a lot of these things. Appropriate justifications for sample size are often missing from animal and translational studies, and sometimes the statistical analytic methods are appropriate, sometimes they're not. Getting a sense as to where all the data is going, not excluding deaths, but reporting on those, and properly using missing data. Adequate CNS penetration. This is really something that is a major part of the translational consideration for preclinical studies. And on our small group today, adequate justification for the proposed dose. What is dose response data in preclinical studies? A lot of times the dose response studies in animals, we would like to see a larger range. Again, and this is something that we can work on as a scientific community. Target engagement. Sometimes in neurology, we do have a good sense as to the target. For other diseases, neuroprotection, in particular, in terms of preventing cerebral ischemia from progressing, there may be a drug may be hitting multiple targets, and it also may be unclear what the exact mechanism of action is. So that's a problem. This is in contrast to, say, a cancer study where we know that a specific part of the cancer biology is deranged and leading to uncontrolled cell growth, and targeting that protein can be very effective, and you can learn that the drug is going to target that protein. And obviously, in some other neurological disorders, the stroke is not the only neurological disorder, we do have a good sense as to what part of the, whether it's the cellular or metabolic mechanism, is the problem, and then if our treatment is actually engaging that target. Pharmacokinetic data, is this in the animal models? Were efficacy studies performed with a route of administration plan for clinic? Again, intraperitoneal maybe is being used in a rat model, and we're not planning on doing that in the humans. Again, it's a parenteral, it's a sort of quasi-parenteral type of administration, but not exactly the same as intravenous, if that would be what we're planning, versus oral administration. And can we get the levels in the plasma and the CSF, if the CSF is an area where we need to be getting the drug, to the levels that they achieved using that route of administration? And did they look at pharmacodynamics? Was there something that actually in the animal studies that can give some link between the biological activity going on from the clinical outcome? And could that pharmacodynamic marker be used in the clinical trial to confirm the idea that the treatment is actually doing what it's supposed to be doing when it moves from the animal to the human?
Great. Thanks, Will. So before we move to all the questions, I'd be interested to hear from Dietrich about your perspective from, your previous experience and now your experiences in industry. If you had any specific.
Yeah, hello everybody. It's great to be back in the CTMC family. We've been all isolated from each other, but we're missing the course this year in person. But it's good to have everybody together here. Rigor transparency, reproducibility. I mean, these are all elements that but it's all about convincing somebody to invest in your research, right? And it's for somebody, it's getting a feel about what are your chances for success, and that's probably a short-term view, because that's do you get the grant? I mean, in industry, it's basically do you have can you convince somebody to to invest a lot of money into trials and programs like that? Same thing as in academia. But in the end, as you're working through developing clinical protocols, it's all about giving your trial, your intervention, the best shot to be successful. And by successful, I'm not certainly also mean that that you achieve the effect, but it's in the end to be safe in patient populations. It's often in clinical research, you don't have as many shots on goal as you have probably in preclinical research. So So you wanna really maximize your chances for success. And even in early phase clinical research, in phase 2 or proof of concept, it's often you're not yet shooting for the big clinically meaningful change across a large patient population, because you might not be powered for that in a small trial. So really want to make sure even to show target engagement, to show that you can move some intermediate endpoint that is somewhat accepted in the field to be indicative of something's happening. There's activity that's happening. That could be success if that's done in a concept of safety. And often in early stage research, especially the regulators, they're really interested in this, really mainly looking at, is it safe? And often these are the two key elements. What's your likelihood for success to achieve the biological effect that hopefully downstream will lead to a clinically meaningful change in that patient population, but at the same time, not to create harm, to make sure that you're giving your intervention the best shot. There are probably two additional points that I want to make based on the video here. The first thing is that as we're talking about that, it's we're thinking about drugs, we're thinking about PK, etc. But I think that's applicable for any type of intervention. May it be a device intervention, may it be an exercise intervention, etc. Same thing. You would have to think about what is the right dose, what's the right way to administer this intervention. If it's a drug or another drug, it's about the how can you actually monitor for that the intervention is being administered as it's supposed to be. With the drug, you do drug accountability. If a subject comes back after a visit and you count the pills and you assume that the patient has taken the drug, you don't know that. So what are your ways to kind of think about to control for that? But same thing is true for so it's just because it's not a pharmaceutical drug, it's maybe a food supplement, it's an intervention, it's a device. Same thing is true here. What are the right doses? What is the right mechanism to administer? Are there any safety concerns? There may be, just because you don't there's not a study that looked at the maximum tolerated dose of a physical therapy intervention. There might be, you might be in your specific patient population. So you have to be careful when you make, or you can think about, inferences that you make from one to the other. Last point that I wanted to make, and so I think in addition to that, also it's a big difference if your drug that you're testing is approved, it's on the market, approved for a different indication, and you take it off the shelf, use it in the dose range. You might have less of a high bar to prove safety, because it's already been proven in a certain patient population, unless your patient population isn't specifically predisposed for more risk here. But it's completely different if it's a complete new molecule that basically your institution or your lab or even your partner in industry came up with, and partnering with you to test this for the first time in a patient population. So different levels of data would be necessary to get through. But in the end, and the final point that I want to make is that I really want to point out what Will said in the video, is that he said, I go back to my colleagues in the lab and talk to them about how they design their animal experiment. I think that's really key. As we're having this clinical trials course, we are the clinical PIs. We are here we're talking about studies or data that has been done or produced before we come in, and often we're recipients, and we can the question is, what if there's a piece missing? What can we do about it? Often, it's after the fact. We need to mitigate. We need to, if we're not sure about the dose, we have to, create more safety monitoring boundaries around that. That's all fine. But in the ideal world, if it's clinical research, in general research and treatment development, this is really a team sport. You want to really team up very early on with your colleagues. They need your input. What is relevant for stroke patients later on? In terms of route of administration. Can they swallow? Probably not. Let's develop an IV formulation or something like that. So I think that interplay with your preclinical colleagues is really important. If you inherit, or if you take an intervention that's been there already, that's been studied widely, be critical with the sources of information that you have. Big issue is publication bias. You only read of the great successes in the literature, and also there you have to take many things with a grain of salt. if a paper is silent on adverse events, don't assume that there weren't as adverse events. If there says no adverse events were observed, don't, it's if you're at the side of the FDA, they don't care about publication. They really look at data sets. So whenever you have the opportunity to collaborate with somebody who did a study previous with you, ask for the data sets, look at it, ask, just because it's not reported doesn't mean there isn't anything or it is safe. Don't assume that. Be critical with what you have in front of you, and ask the question, what do you need to know, starting planning your protocol for your intervention.
Yeah, I think, and one of the points that you make that I wanted to reiterate is that, for clinical trials, we don't get as many shots, right? I think that was a great statement. You really want to make sure that you have, and not necessarily because it's harder to get them funded, but you're also using people's, you're using human subjects, and you may be working in an area of a rare disease where once you try whatever it is you're trying, those people aren't eligible for other trials, depending on what it is. So I think, and the cost of clinical trials are more expensive for sure than animal studies. So I think the more care you can take prior to starting your clinical study, and that's why the importance of rigor, the better. I think we can go to some of the questions that are there already. We can start with, there was a question about pharmacokinetic studies and how do you approach possible variation attributed to intervention that's not already well established in the literature. I'm not sure I completely understand that question. No.
Maybe
Do we want to put does Dr. Choi have his speaker on? Maybe he could ask a clarifying question if we give him. I think you can invite him to talk, right, Joy? I think I can too, maybe. Maybe we can't. Maybe we've been, no, there should be a way to allow the talk.
Yeah, it's under participants. Hold on one second.
I think I got it.
Oh, okay, thanks.
I assume, hello.
Oh, hi. Well, thanks for looking at my question. Well, to clarify, I guess, the reason I was asking is it's not characterizing the pharmacokinetics of a drug, but seeing if there are changes in the pharmacokinetics due to it's a diet intervention. It's a ketogenic diet, and there happens to be not much of anything that I could find so far on pharmacokinetics in general in ketogenic diet, but specifically levodopa or kind of dopaminergic medications. So I guess in terms of, this came up in review of the protocol, that it wasn't being accounted for. That, holding the dose, not holding the dopamine dose, but holding other medications and giving the same dose schedule, testing schedule, but still doesn't address, does the diet, could it affect the absorption, the levels? So it's a very I think valid question, and it seems like there's different ways to go about pharmacokinetic study, but just wonder if that comes up, that because the purpose of the study is not to to really delve into pharmacokinetics, which are in general they're pretty well established for levodopa. So I guess specifically the thought so far was just to keep the same blood draw schedule. So not doing like an additional series of blood draws to define pharmacokinetics, but rather, right now tentatively there's AM and PM blood draws. So just adding on, whichever, I guess levodopa, probably make the most sense to test that. And then it could be done pre and post diet intervention to see within participants on the diet, you know, is there a change in the levels? But yeah, I mean, this is getting into detail, but I guess it's just like, is this valid? It's not going to be as detailed or as accurate as, you know, really measuring, I don't know, say 6 times, 8 times, you know, every 30 minutes. I mean, of course, that would be the ideal way to assess it. I mean, that's still a possibility, but so far that wasn't the kind of the tentative plan to you know, to add on blood draws and, you know, a little bit of challenge, I mean, technical challenge to get that done correctly.
So you're talking about how the diet might affect the pharmacokinetics of the other drugs, right?
Right, exactly.
Yeah.
And just because there's not a literature, it'd be nice if I could just turn to literature and say, this is what, you know, this literature shows, but just it hasn't really been investigated, it seems.
Yeah, it is complicated. And I think that's true in ketogenic diet in general. I don't know, Dietrich, did you want to comment at all on this, or I can?
Yeah, I think again, it's all about, you know, controlling for potential confounds, and I think these are really valid questions early on, you know, in development. Is you know, especially if you have something that is relatively established and well known, such as the pharmacokinetics of L-DOPA. And if you want to downstream, you know, assess, you know, effects of a dietary intervention, you know, may it be due to, you know, change of resorption of or the pharmaco, you know, kinetics of L-DOPA. That's a relevant question. That might not be your main question for your study, but that's, you know, a potential confound. You know, this is especially as you build up your case in your early stage study for, you know to create, you know, enough pilot data to then, you know, follow up with a bigger trial on the clinical effects. You know, these are often good additional, you know, data points to have to make your case that, you know, it is not affecting, you know, the pharmacokinetics of L-DOPA, or it may be. So I think it's a matter of, you know, often you know, for that it can make things complicated. Because, you know, you need to have an assay, you need to think about, you know, the logistics around that. I mean, one thing that I learned in industry is, you know, what I learned at NIH and at NINDS and the CTMC is that you know, there have to be two, you know, people developing your clinical trial, that's you as a clinician and your partner in crime, the biostatistician. In industry, what I learned is that, you know, a third person actually comes to that team, and that's a clinical pharmacologist. That really, you know, because these guys do magic, you know, in terms of modeling. You know, they with sparse sampling and data points, they can give you a good, you know, amount of you know, modeling and data that can help you, you know, later on, you know, have more confidence about your dose and have more confidence about that, and elements like that to include. If you get a review like that, comment like that, it's often comes out of left field, but it's, I mean, I have done, you know, PK sampling at academic studies before, and it's a little bit of a hassle, but I think it adds, you know, significant value to a study.
And you know, another thing to just sort of generally consider is, you're right. Sometimes the understanding may be so sparse that you don't really know what to do. And where do you start there? Sometimes the answer is there is some animal data that would give you at least a starting point, knowing that animal metabolism is not the same as human metabolism in all cases. And sometimes the answer is, if there's not really even any animal data to calibrate, if there are reasonable metabolic models in animals, sometimes you know, we think of translation as one way, but it's got to be two ways, right? Like we find uncertainties in humans, or we find a trial that didn't work. We have to continue to go back to the translational colleagues and say, oh, let's tweak this, or we need to know more about this or that. And I think a lot of this has to be the sort of two-way street of things. We're not merely consumers of preclinical data. We have to, we're sort of the market, right? And we got to be like, hey, I
Yeah.
want to have a different color Tesla, you know, like I don't just want white. I want a, you know, lime green Tesla. Maybe I don't really want a lime green Tesla, but you know, but I think, you know, to get to some of the other questions, Laurie, I mean, we should probably cover that. Did you want to start to cover that social science question? Because I think that's a great question in terms of how things are handled and thought of at NIH with respect to areas of research in which, you know, there isn't necessarily a quote unquote animal model.
Yeah, I think, you know, there's a question about how do you deal with the multifactorial social and environmental effects? I don't know.
Well, I guess one of the, oh, I'm sorry, I interrupt. You go first.
Said, I was going to say you gave some good references for this, but go ahead.
Yeah, I was just going to add on in that sometimes, particularly in that form of research, in terms of maybe you're doing behavioral interventions, there are theories of behavior, and there's empirical social science research that undergirds that. But I think when you are citing that, the degree to which it's well accepted in the field, what sorts of both empirical and experimental research went into helping to develop those theories, can help you give that sort of supportive case as to, I'm not just using my intuition to say, oh, I think I can get people to reduce their blood pressure by telling them that, you know, they'll be better people if they have lower blood pressure, right? Like using, you know, that's sort of non-motivational, like, let's just try this. But no, like, what is the social science theory? Is there a theory of planned behavior? How do you get people to start to make behavior change? So I think keeping things oriented towards theory, prior empirical research in the social sciences can be crucial, but then also thinking about whether things were done experimentally in social science studies. And it can be challenging. It's a little different. You know, human behavior is, I mean, brain ischemia is complicated. I mean, human behavior is really complicated. So I think it's important to think of those things.
So before we run out of time, there are a couple of other things that I think we should probably address that we touched on. One comes from another question that came in about using an intervention for in one disease that has only been used in another disease before, and how much preclinical data do you need if you're going to switch diseases for the drug. And I guess one of the other questions that I would also like us to talk about is the importance of how important is it to show that you've got target engagement, right? Which, but so let's talk about the first one. I think, you know, at least at NINDS, you're gonna need to show, let's say, so the question was specifically about an ALS, using a drug in ALS that hasn't been used in ALS before, and do you need to show that it changes, and there's some change in the animal model for ALS, or do you not need that? And I think most of the time you're going to need to show that at least it has the same effect in your disease or a comparable disease area. I don't know if Robin wants to comment as well, but you could also get some data by some at least observational data, and then, like we talked about, go back to see, you know, go backwards and take your observational data backwards. I don't know if you want to say anything about that, Robin or Dietrich, either one. Will?
Probably what I can say. I think it's. I mean, the question is, what do you need? Who do you need for? Who do you need to make a case to? And these are usually your stakeholders that want to invest in you, and that's you know, your funding agency, right? And they have, you know, 15 grants, and then they look or grant proposals, and if you know, if you rank, you know, the potential impact, you know, you can already imagine that, you know, whatever is written in the rationale or in the justification section there convinces the reviewer to say, okay, good, there's you know, this has a higher likelihood for success, or there. I mean, think about, you know, your levels of evidence, right? The lowest level of evidence is, you know, eminence based. You know, basically you as an expert in the disease saying it will work because I say it will work. But you know, the highest level of evidence is that you really have, you know, actual data that is well controlled, and there. So it's the same thing here as if you try to get a good paper published. So the better the case you make, the better the data you have at hand, or you know, either you produce them yourself, or you know, you, as Laurie says, or if it's a drug that's approved, you know, if there's a way to tap into, you know, big, you know, registries, you know, think about, you know, some retrospective, you know, analysis where you might, you know, get a trend somewhere that you know, that could help make a case. Obviously, you know, the higher the level of evidence, and again, this comes back to the NINDS recommendations, that will be the gold standard. If there are adequate animal models that are really predictive of success, and you have, you know, well-controlled experiments there that show the effect, it's obviously great and it's obviously good. But you know, knowingly in CNS, you know, sometimes, you know, our animal models are not that perfect. It's not that case that they are not there. I mean, even if it's an animal model that just shows that it goes into the brain, that's better than no animal model. So and often, you know, animal research can be quite, you know, non-specific. You know, it just reduces, you know, the agility of the rats without, you know, anything specific. Again, that's better than nothing. If in your particular disease, you know, models are, you know, the models are not that perfect. Maybe the last point is that if a drug is already approved and on the market for a different indication, often, you know, that helps you. You know, look at the label. You know, the FDA publishes all the labels, even the reviews when they put out a drug on the market. That really can help guide you. You know, what is a safe, you know, dose range? What is a you know, what is a maximum tolerated dose, etc. If you you know, that, you know, has also regulatory implications. So, you know, who else do you need to submit applications to? But that's usually a faster track, and it helps you guide to your experiment also what you have to monitor for. If you are getting your drug from a pharma company, for example, either it's approved or not, ask them. Often these data are sometimes not in the public domain. But you know, if you get your drug from a company, make yourself familiar with a very important document. It's called the investigator's brochure. Usually, everything is written in there that has been done, and that's often a great, you know, set of reference for studies that you just don't find on PubMed or even on clinicaltrials.gov. So,
and I guess I would just add, in terms of this ALS idea, it has the mechanism, the purported mechanism really matters, right? Like, so if you think that if cancer cachexia or decreased caloric intake in cancer is felt to be mediated by the same sort of causal pathways as it is in ALS, I think you're going to have a stronger argument. However, if you think that it has some different off-target effect that's going to maybe modify mitochondria or something like that, then you definitely are going to need an ALS model to sort of say, this is actually hitting the mitochondria, this is doing this is slowing the progression of ALS. If it's a matter of, you know, you could possibly get information about caloric intake, you know, muscle stores, glycogen stores, and so forth. You know, if the mechanism is thought to be similar and conserved across ALS and cancer, but if it's sort of, you think it's something that's going to be novel, you know, I think having the data in that disease is going to be helpful. And I think if there's a reason to think that has promise because of other, you know, somebody has to have some reason as to why you think it might help in ALS. If it's just to improve appetite, you know, that's great. You know, that there's different, you know, knowing what the mechanism of appetite increase is helpful. And if you know that this can affect, you know, cross into the brain and stimulate appetite, and that people will eat more, if we know that people with ALS who eat more do better, then, you know, it may be, you maybe don't need the specific animal model in this case. But if it's like, I think it's going to start to do other things, you really have to have a strong justification as to what those other things are, and if it has been able to modify the disease in ALS.
Yeah, Robin's point in the chat box is that mechanism is often hypothesis driven, and whether or not you think it's the same mechanism, you'll need the data in the same disease. And that's sort of a concise way of saying what I think we've all been trying to say. I also think that, you know, it depends on the amount of, again, to go back to the observation and then taking it backwards, is the amount of clinical effect that you see. If you see something, you know, that has a significant clinical effect, then it may be easier to go back and design the preclinical, exactly what you're looking for in the preclinical studies. Yeah, and if the other point is, if you go back and watch the webinar video, we discussed the ALS lithium question, and those of you who may remember that lithium came out after some preclinical data that lacked some rigor, but drove a significant clinical trial adaptive trial in ALS, to try to see if it made a big difference, and it turned out to be futile. So,
well, one point that I'll make related to rigor from the statistician side, because we're often the cranky fuss buckets of the clinical research world, but preclinical data that are leading interest in a clinical study are almost by definition biased in a positive manner to be stronger than they probably actually are, because you're only doing the study because you like the results from the preclinical data. So some dose of skepticism is always needed in some sense, because even if it's, you know, if you had done a preclinical study and didn't like the results, you wouldn't even be talking about the next study. And so some of those are going to be true, but some of them could just be type 1 errors preclinical. So I think you always have to, and that's why you really need strong evidence to really support that, to make sure you can tell, because there's looking forward and looking back. Everybody's convinced it's going to work, but looking back, if you don't have strong data, you know, the field, especially in neurology, is littered with failed studies that, you know, that everyone could make a strong argument for up front that just didn't pan out in the end.
Yeah, and I think other things can help provide collateral information that helps strengthen that evidence, whether things were tested multiple, you know, like these tend to, you know, again, not everything is stroke, but you think about the STAIR criteria, multiple doses, multiple severities, multiple, you know, small animal models, large animal models, behavioral testing, in addition to, you know, volume of infarct, you know, enough things need to be going to make sure that you've robustly tested the sort of boundaries of where this intervention works and where it stops working, can help improve confidence. But as Chris points out, you know, we would like there to be more of the unsuccessful preclinical studies published, but that we know that it's harder in that literature currently, although hopefully that's changing. In the clinical trials literature, we obviously make a big point of making sure that our neutral studies are getting out there, but it is less the case in the preclinical world, and we have to work to try to help people get more of their things out there, so that people are looking at it in a more nuanced way.
well, we don't have, we have like 3 minutes left. I'm not sure that we can address too many other questions. I guess one thing is, what do you do if there's no agreed upon animal models? I just am curious to hear what you think about that. If there's really
I asked Dietrich what to do.
Sorry, Dietrich, we invited you and now we ask you this.
Come up with one. No, I mean, I think you know, animal. Okay.
Well, I guess. So, go ahead.
I think my point is that as you know, animal research is important not only for your efficacy, but also for safety. So some idea that you can draw from the literature or from preclinical data that is not necessarily an animal model of your disease will still be helpful, kind of guide you in which range you should look. Okay, and there is an argument to be made if that's even if you think about biologics, for example, if you have a disease that has only specific epitope expression that you can't really be guided by an analogous model, that there is value to argue that it can only be studied in humans. That's the rare disease. It has only that epitope. There's a strong. You can make an argument with if you have a good idea about the dose range that you're exploring. There's a high likelihood based on even in vitro data that you're hitting a mechanism there, and there's a way even clinically. I mean, back to Alex Joyce's question, is that even if your hypothesis is you engage a certain pathway and you can measure elements of a pathway in a pharmacodynamic way, and you do that in humans and you collect additional clinical information, that might be good enough.
Right. So thanks to everyone who's joined, and especially thanks to Dietrich for helping us out with this, with your great perspective. And Joy just posted an evaluation link. If you could please fill this out, that would be very helpful to us. And I think.
And I just wanted to make sure to let everyone know that our next webinar is July 21st on what to know about sample size. So stay tuned for an announcement coming out in the next week.
Great. Okay, thank you guys very much, and stay cool.
That was fun.
Thank you.
All right.
Bye.
Thank you.
Bye. Good to see you, Dietrich.
To see everybody.